Hey, welcome! Did you know that inside every single one of your cells, there's a ridiculously detailed instruction manual? It's a master blueprint that decides everything, from your eye color to how you fight off a cold. Today, we are going to unlock the secrets of that blueprint and explore the molecular language that writes the story of you, me, and every living thing. I mean, just stop and think about that for a second. It's a wild question, right? How does life pull off this incredible construction project, starting with just one tiny cell? Well, the answer is hidden in a remarkable molecular language, and together, we're going to learn how to read it.
So here's how we're going to tackle this. First, we'll start with the master blueprint itself, DNA. Then, we'll check out how its messages get carried around and turned into the proteins that do all the heavy lifting. We'll also see how the whole process is controlled, and finally, we'll explore how figuring all this out is literally changing the world. Okay, let's kick things off with the most famous molecule in all of biology, the true architect of life, DNA.
So what exactly is DNA? Well, it's this long, two-stranded molecule that's twisted up into that classic double helix shape, and its main job? Information storage. Seriously, think of it like a biological hard drive. It's built to safely and very stably protect the genetic instructions an organism needs to develop, live, and reproduce. Now, this is where it gets really clever. Let's compare DNA to its cousin, RNA. See, DNA's double helix structure makes it super stable, which is perfect for storing the master plans long-term. RNA, on the other hand, is just a single strand, so it's less stable. That actually makes it perfect for its job as a temporary messenger. And notice that little chemical difference? DNA uses thymine, or T, but RNA swaps it out for something called uracil, or U. That tiny change is like a secret password that lets the cell's machinery instantly know if it's dealing with a master blueprint or just a temporary memo. So if DNA is the master blueprint and it's locked away for safekeeping in the cell's nucleus, how do the workers out on the factory floor actually get their instructions? Well, that's where our messenger, RNA, steps in.
This whole process is called transcription. And honestly, the best way to think about it is like making a quick photocopy of one page from that giant blueprint. First, a little section of the DNA double helix unwinds and opens up. Then, a piece of cellular machinery, this enzyme called RNA polymerase, zips along one of the strands, reading it and building a matching single-stranded RNA copy. This copy can now safely leave the nucleus and take the instructions out to the rest of the cell.
But hold on. That first draft of the RNA message isn't quite ready to go yet. In complex cells, like ours, the genetic instructions are full of these, junk sections called introns. So the cell has to do this brilliant editing job called splicing. Think of it like editing a movie. The introns are all the outtakes and bloopers that get cut out, while the important parts, the exons, get stitched together to make the final coherent film.
All right, now that we have our perfectly edited RNA message, we are ready for the main event, taking that code to the assembly line and building an actual protein.
You see, the RNA message is written in a language that only has 4 letters: A, U, C, and G. The cell's protein building factories read this message in 3-letter words called codons. Each codon is a specific instruction for one particular amino acid, which is the basic building block of a protein. So the sequence of these codons tells the cell exactly what sequence of amino acids to use.
This assembly process is called translation. It kicks off when a little molecular machine, a ribosome, clamps onto that messenger RNA, or mRNA. As the ribosome scoots along the message, another type of RNA, called transfer RNA or tRNA, acts like a delivery truck. It reads the 3-letter codon and brings over the exact amino acid that was ordered. The ribosome then links these amino acids together, one after another, building a chain until it hits a stop codon. That's the signal that the protein is finished, and it gets released. And the final product? A fully functional protein. We're talking about everything from the enzymes that digest your lunch to the hemoglobin carrying oxygen in your blood. These brand new molecules are the tireless workers that perform almost every single task needed for life.
Okay, so the cell knows how to make any protein it has the code for. But let's be real, it doesn't need every single protein all the time. So how does it control which genes are turned on and which ones are kept quiet? It turns out the cell has genetic switches. In bacteria, for example, genes that are all needed for the same job are often grouped together in a handy little unit called an operon. This is super efficient because it means the cell can use a single switch to turn an entire production line on or off all at once. A classic example of this is the lock operon in E. coli bacteria. This operon holds all the genes for the enzymes that break down lactose, which is a type of sugar. Normally, these genes are switched off to save energy. But if lactose shows up, it acts like a key that flips the switch, turning the genes on so the bacterium can start using that sugar as food. It's a beautifully simple and elegant system. Now, while bacteria use these operons, more complex critters like us have a much more sophisticated network of switches that control each gene one by one.
Now, understanding these tiny molecular processes has allowed us to do way more than just look at cells under a microscope. It has given us the power to read the book of life itself, and the consequences have been absolutely profound.
You've probably heard of the Human Genome Project. It was this massive global undertaking to read the entire human genetic blueprint, all 3 billion letters of it. This gave us the first complete look at our own instruction manual, and it led to incredible breakthroughs like identifying the BRCA genes that help us understand a person's inherited risk for breast cancer. And one of the most fascinating things we learned is that while our DNA is over 99% identical from person to person, there are these tiny little variations in the code that make each of us unique. Scientists have a name for these variations, polymorphisms. And DNA fingerprinting is just a technique that zooms in on these specific differences, creating a unique genetic profile that can identify a specific individual.
The real-world applications are just mind-blowing. In forensics, it can put a suspect at a crime scene or, just as importantly, prove someone's innocence. In conservation, it helps us protect the genetic diversity of endangered species. It lets us trace our own evolutionary family tree back thousands of years. And, of course, it is completely revolutionizing medicine, leading us toward a future of therapies that are tailored to your specific genetic code.
So, let's step back and bring this all together. What we've just walked through is the absolute core principle of molecular biology, something often called the central dogma. It's the fundamental flow of all genetic information. Information is stored long-term in stable DNA. Through transcription, a temporary RNA copy gets made. That's our photocopy of the blueprint. That copy is then read through translation to build a functional protein, our machine. And the whole factory is carefully controlled by regulatory switches. DNA to RNA to protein. That, right there, is the fundamental story of life at the molecular level.
We've gone from just observing life to being able to read its source code. The power this gives us is absolutely immense, and the ethical questions that come with it are just as big. As we stand on the edge of a completely new era in genetics, the question isn't just what can we do anymore, it's what should we do. What comes next? Well, that's up to us to decide.